Hi students, today we are going to talk about the topic mutations. Mutations are quite evident to every one of us. Even the small uh, kids that we come across today in our day-to-day life say that, oh, of course, mutations are possible because mutations are quite familiar to every one of us. But there was a time when mutations were quite unpopular. During the 1890s, the concept of genes was being discovered by Hugo de Vries, a scientist, and he put forward the laws of heredity while apparently unaware of Gregor Mendel's work for introducing the term mutation and for developing a mutation theory of evolution. This man was a scientist who studied on Enothera lamakiana, the evening primrose plant, and he found that some of the variants of this particular plant were being available, and he suggested that these new plants were mainly being available not because of due to accumulated evolutionary factors, but it was mainly due to the presence of genes which undergo certain mutations rapidly. And had mutations as a sudden changes of any behavior. Okay, so he is known as the one who suggested the term mutation, and Hugo de Vries is well known for his mutation theory. So let's go into that. Okay, he told about one plant, and right now, if you go across the world, you can find that there are different types of mutations which are possible. If you go to see into animals, there are different now. The frog which I have showed you, it's a three-headed frog. There are two-headed cows. There are reptiles which are having limbs. There are different animals with different mutations possible. But at the same time, we should remember that not all animals do have these mutations. Not all frogs are being found to be three-headed. These occurrence of mutations are being found to be very rare, and it is mainly due to some rapid changes which occur during their development. Now, if you go to have look at the plant where I photo which I have shown here, that it's coming from the Michigan State University. They are, you can see even the petal itself. Half the plant is having one color, and the half the plant is having another pigmentation. Coming to the leaf variety which they have shown, some of the leaves are having a green pigmentation in the middle, but some other leaves are having a different type of pigmentation pattern. So mutations are things which happen in nature as a sudden change to something. Even in the orange variety that you have here, you have a dark orangish color for half of the melon, but half of it is having a yellowish tinge. So that's the case of plant and animal mutations. And so much is the influence of mutation that a fictional story on mutation called the X-Men is also quite popular right now. So we also imagine of people, of human beings, probably those who are having mutated and are who are having different powers could be located at different parts of the world. Now, of course, that's quite a fictional story. Uh, and we'll see on to the basis of mutation right now. Mutations are discovered, they are usually defined as sudden heritable changes which happen, and these changes can happen either in the genes or in the chromosomes. And when they happen in the genes, they are referred to as the gene mutations, and when they happen in the chromosomes, they are referred to as the chromosomal mutations. Gene mutations can happen by either the replacement of one nucleotide by another, or the addition or deletion of a nucleotides. So genes are made up of nucleotides, and changes in the nucleotides would contribute to their gene mutations. Now coming to the chromosomal mutations, it could be due to the change in the number of the chromosomes. that comes to polyploidy, aneuploidy, you'll be learning in details. And it could also be to the change in the structure of the chromosomes. So, when you talk about mutations, if you are talking of prokaryotes, the level to which the gene mutations can happen sometimes may be limited to point mutations. It's not necessary that you can put a demarcation that they would only have this, because mutation as such itself is a sudden heritable change. So things could happen vice versa too. Coming to humans, of course, we have both the type of mutations, either gene mutations as well as chromosomal mutations also. And let's go into the details of this. Gene mutations can either lead to point mutations, okay, or they can be referred to as large mutations. Point mutations usually affect only one base pair in a given location, and the alteration of a single pair of nucleotide, and it mainly occurs by the addition or deletion of one nucleotide pair in the coding region of a gene. Whereas large mutations are less common, and they mainly include large insertions, deletions, inversions, duplications, and translocations of a nucleotide sequence. So in the point mutations that we have, you have a single nucleotide might be changed, added or deleted. But in the nucleotide sequences itself, sometimes instead of having a single nucleotide change, a large stretch of a nucleotide sequence might be inserted, deleted, inverted, duplicated, and also translocated. The details of which will be quite evident as you finish this lesson. So let's see what is a point mutation. In a point mutation, you can see this is the original sequence now. This is the original sequence. And what happens, instead of the T here, it has been replaced by a cytosine. So this would result in a change in one base in the DNA sequence. Okay. Now coming to insertion. In the insertion here, see you have TAACT. Okay. And then G. Here TAACCT. That is, one base is being inserted inside into the sequence. So that is what is a insertion. Coming to the next one is okay, we'll talk about deletion, then we'll go to the other one. You have here TAACTG. Here what happens? TAAC and the T is being removed over here. Okay, and coming to the next one, the substitutional type of mutation, you have here CTG, okay, not CTG, TGC, okay, TGC is being formed, is the one which we form here. See, that is 1, 2, 3, 4. After the fourth one, we have TG and C. Okay, instead of instead of TGC, another codon called the what? GA and T is being replaced. So, one or more base pairs are being replaced by the same number of bases in the case of substitution. Another thing in this picture, what we need to note is that, now we all know that, okay, genes code for certain proteins. And these genes are being read in the case of, in the form of codons. That is, every 3 nucleotide base will code for an amino acid. Now in the first case as well as in the second case, that is in point mutation and as well as in substitution, okay, you see, if you're going to consider this as a start of a gene, you have TAA. I'm reading about the codon. TAA is a codon, CCG is a codon, CAG is a codon, and then you have GT and another one as the next codon. So if you go to divide them into 3, what happens, you can see that though the bases are being changed, okay, no change in the reading frame of the codon is happening. Same is the case with, is this the same with, now if you have TAA, here CGA, TAG and GT, okay, here if this is the start, you have TAA, that is read like that, CTG, this is another one, CAG, here also there is, what is it? There is the shifting of the frame of this thing is not happening. But if you go to come here, sometimes the genetic codon itself, the frame of that would be shifted. And that is resulting in the case of mutations called the frameshift mutations. Okay. In some cases, what happens, the shifting of the nucleotide DNA codon frame would happen. So that is when the change in the nucleotide base will result in the shift of the codon, or the change in the reading frame of the codon. That is referred to as the frameshift mutations. Another one that we have here is the inversion sequence. Okay, it's just one particular sequence here. Now see, TGCA is just inverted and it is reversed. So that is a inversion. So we'll talk about the remaining in the next part. Thank you.